Have you ever wondered what would happen if cell division did not occur? Well, for starters, you wouldn't even be here right now. Interestingly, a new cell division mechanism has been discovered. But first, let's start with some context. As we know, mitosis is the process of nuclear division that results in two daughter nuclei genetically identical to the parent nucleus. Subsequently, cytokinesis, or the division of the cytoplasm, results in two daughter cells. But what exactly happens during cell division? How are the chromosomes pulled apart? We know that microtubules, long tubular fibers about 25 nanometers in diameter, are involved in pulling chromosomes to the opposite poles of the cell. However, do chromosomes also play a role in cell division? Recall that chromosomes are gene-carrying structures consisting of single long molecules of double-stranded DNA. It was long believed that chromosomes played a passive role, that they were simply pulled by microtubules and did not affect cytokinesis. However, it was recently found that chromosomes do play an active role in cell division. How? Let's backtrack. It all starts with the cell cortex, a specialized layer of cytoplasmic protein on the interface of the plasma membrane. This can aid with cell shape for animal cells, which do not have a cell wall. British and Canadian researchers wanted to test if animal cells assemble a rigid cell cortex before mitosis, and if so, whether or not the relaxation of the cortex at opposing cell poles contributes to cell division. To test this idea, the researchers analyzed sensory organ precursor SOP cells in the Drosophila flies. Live cell imaging was used to study the changes in cell shape during mitosis, as presented here. Furrow formation refers to the groove formed in the cell membrane of a dividing cell. In both the fly cells and human cells, actin, the material which comprises the cell cortex, was not present at the cell poles. This means that relaxation of the poles of the cell occurred before furrow formation. Anaphase cell elongation was also independent of furrow formation. As you can see in this graph, most of the cell elongation actually occurred before furrow initiation. This data suggested the existence of a second parallel signaling pathway, which triggers the relaxation of the polar cell cortex at mid-anaphase. This is different from the pathway by which microtubules pull apart the chromosomes. So what exactly is the source of this parallel pathway? One of the key signals researchers identified was an enzyme complex, a phosphatase known as SDS22PP1, which is found at the kinetochores of the chromosome. So back to the original question, how do chromosomes play an active role in cell division? The enzyme allows for a signal to be emitted by the chromosome. The signal emission contributes to the softening of the polar membrane, facilitating the elongation of the cell and the following division that occurs at the equator. This means that the chromosomes themselves are actually reinforcing the microtubule action. Now, what is the significance of the researchers' findings? The discovery of this mechanism adds to our knowledge of cell division. This is important as cell division is essential to life. This is an astounding development in the basics of biology, changing our ideas about the very concepts we thought we knew. The research illustrates the existence of a pathway not typically mentioned in the average textbook. It is built upon the foundation we have learned in introductory biology, including our understanding of mitosis and how the microtubules pull apart the chromosomes during anaphase. The research also adds to our understanding of how enzymes facilitate life processes, in this case, cell division. These findings can be applied to a large range of issues in the scientific world. Specifically, cancers are characterized by abnormal cell division. Thus, this process is looked at as a target to prevent cancer. Expanding our knowledge of cell division may aid in creating new therapies for inhibiting the division of cancer cells without affecting healthy cells. A revolutionary change that all starts with one cell.